of the network, and you're going to look at interaction between brain regions. And this effective connectivity can be estimated to get signature of the brain dynamics. So you're going to talk about decoding of conditions, pathological conditions, or cognitive conditions. And we're going to use that to derive some biomarkers and things like that. In the second part of the talk, I will see how the same model can be used this time to interpret the brain dynamics in terms of network complexity. Okay. And then in the end, I will go back to different flavors of whole brain modeling, anatomofunctional, so model that can combine anatomical data like structural connectivity and functional data, and see how we plan to develop to extend this model in the future. Okay, so the first thing that I wanted to talk to you about is a recent paper, well, not so recent now, where we had some stroke data from the group of Matthieu beta, and this was in the context of the Human Brain Project. And that data set, sorry, what we had is a number of patients, 130 here, and a few controls. And the specificity of this data set is that at 3 time points, we had the patient scanned and evaluated. So at 3 weeks, just after the acute phase, after stroke, 3 months and 1 year. So they had both structural and functional scans in the MRI. And the clinical evaluation involves some cognitive tasks, so to measure their deficits in different domains like motor, memory, etc. And so what has been already shown by the group of Matthieu is that the diffusion MRI, so something that you see here, where you look at, for one patient, where you've got a lesion and how it affects the structural connectivity, so the interaction, the anatomical pathways between the different brain regions. So how this can predict the recovery of the patient. So trying to understand, to predict how many deficits, cognitive deficits they will have at 1 year. And so our question was to understand how functional MRI could contribute to this prediction. So when you get fMRI signals like this one, what you can do is to parcellate the brain, meaning dividing it in a number of regions. Then you've got time series. You can apply a measure on that, and you get metrics like the functional connectivity here. And you get one for each patient, and you can do your prediction for cognitive states or pathological states, like here, the number of cognitive deficits. And so what we tried to compare that is a model-based approach. So instead of applying directly a measure, we use a model here. So a model where you've got here just 4 regions represented, but in fact, for this model, we had like 300 regions for this data set. And we evaluated the interaction, so that's the effective connectivity. That's basically the strengths of how one region influences another in the model. And so you can estimate this weight. So you've got a large number of parameters to estimate for each session, which is challenging. But you've got method for that. And so you can use this effective connectivity matrix, so all these matrix of weight, you can vectorize it and use it in your favorite classifier for your prediction. The second part of this model, second aspect, is to constrain the topology of this network. So you see that here you don't have an interaction between these 2 regions here. And that comes from the structural connectivity. So we get DTI data, we look at how anatomical pathways connect the different regions, and we only put in the model the interaction that we see in the structural data. Okay. So that's anatomically informed network dynamics. And so using this, we can also incorporate information from the connectome, like when some patient, they've got a damaged pathway, we can include that in the model. So we end up with a number of features. That's the vectorized effective connectivity or functional connectivity for each line. So each sample here is one subject. And we're going to classify them into the number of deficits that they have. So here the label, if they've got 0, 1, or 2 deficits or more. And we use modern classification techniques like with cross-validation and so on to do this prediction. So why we need to use this technique is that it's well known in fMRI that you've got different sources of variability, and one of these is subject bias. So if you look at, this is another data set, so you've got 6 subjects here. They've been scanned like 40 to 50 times each. And if you look at the similarity between the fMRI or effective connectivity or functional connectivity across the sessions, you see that you've got higher similarity here in the blocks for the session of the same subject compared to the rest. So you've got a bias that you want to remove. And so if you want to find classification for condition, like here the pathological condition, you want it to be somehow orthogonal to the bias for the subject. So if you manage to orthogonalize the signature somehow related to the pathology and the subject, then it's fine. But if you don't do that, then in the end, you've got somehow the subject identity that's contaminating the condition signature. So long story short, we applied this method here to try to extract and to predict the cognitive scores. And so what we do when we repeat several splits of the data in a train test cross-validated fashion. So we get a distribution of accuracies here for our functional connectivity. And to understand if the prediction is good, then we use permutation of the labels to get a baseline distribution of accuracy. So as you can see, the variability of this baseline is quite large. And so the point is to see how better we do compared to the chance level. So with functional connectivity, we do better than chance, and we're happy. But when we use this effective connectivity, then we do even better. And so we increase a bit the prediction when we use this effective connectivity that captures not only the spatial correlation of the fMRI, but also the temporal correlation to some extent. So we are happy. And the second thing that we could do with this technique is in our space of features. So we've got all the different interactions. In the classifier, we can identify the one that contributes to the good prediction. And so when we represent them on the brain, we can see a lot of interhemispheric interactions. So these predict the reconfiguration of the brain that support the recovery or not of the cognitive deficits. And so the interesting point here is that we showed that they're not just close to the lesion, but it's very widespread reconfiguration of this interaction across the brain. And the disappointing thing in this study was that we tried to combine the functional data with the structural data to see whether there was synergy between them for the prediction, and the answer was negative. So what I show you is how to use this framework. And since I was asked to talk more about the method, so I tried to convey my enthusiasm for mathematics with some equations. So bear with me for just 5-10 minutes. So just to present you what is behind this. So it's in fact a simple dynamic process. So it's the Einstein-Uhlenbeck. So you've got the derivative of this activity for each region that's corresponding to an exponential decay governed by this time scale tau. So something that we saw already yesterday talking about the time scale. But this model is a network model. So there you've got the interaction So that's the effective connectivity coming from the other region. And on top of that, each region is receiving some input that is kind of noisy input. It can be thought of intrinsic noise or for sensory areas, some stimulus-related inputs. So what's good about this model is you've got two sets of parameters, this Jacobian corresponding to the exponential decay and the connectivity combined together, and the covariance of the inputs. And when you analyze this model, what you should look at is the covariance of this activity. So these parameters here, they determine in a unique fashion, so you've got a one-to-one mapping between them and the covariances. But for that, you need to look not only at the zero-lag covariance, so covariance when you take two time series, like the blue and the gray here, but also when you shift one time series, so from blue to green, to look at the covariance, and that's capturing the propagation nature of the signal. You can see here, for example, with the blue, you've got a peak between these two regions, but when you shift that, that's changing. So that's somehow describing if some regions they are ahead compared to others, but looking at this phenomenon at the scale of the whole network. So the important thing here is to capture the network effect. Okay, so you end up with these matrices of covariance without time shift and with time shift. And the point then is to match the model covariances to the empirical data. And for that, you can use analytical equations that describe, as I said, these matrices from the model parameters. And what we design is an optimization method that is a gradient descent, in fact. Sorry. That is just at each time step, looking at the model error, so the difference in these matrices between data and model, and calculating the update. So here I just show the example of the update for the Jacobian. You've got the same for the input covariance. Okay, so just to show that it works, in the end you can map your empirical data to the model. So here is the functional connectivity. And you can do it unconstrained with the structural connectivity or with the constraint. So here's an example without the constraint by the structural connectivity. And the important thing also is that to measure the network effect, so if you look for each interaction, so each pair of regions in this network, here, so here you've got the interaction, the EC weight, and here you've got the correlation or the correlation there. So you don't have a trivial mapping between them. So to generate the correlation pattern at the network level, you really have a network effect, and it's just not that two regions, they are strongly connected, resulting in strong connection between them. So, all right, so I hope the math were not too heavy at this time. We'll have just a few equations more later. But that was important to show you a bit this model to later understand how we can use it for interpretation. And in particular, interpretation for in terms of network complexity. So it's a simple model, but the cool thing is we can, in a way, capture the heterogeneity in the data, in the fMRI signals. And so to show that, I'm going to talk about a different data set that we analyzed from the group of Stephen Lorries in Liège. And their group, they're very interested in different states of consciousness. So consciousness is very nice and dangerous field because you've got all sorts of creative work about that. So that's fun to work with people there. And somehow you've got two side of consciousness, so one related to wakefulness as compared to sleep or coma, and the other with the content, so the perception of stimuli. And as you can see, they are related, but the nice thing is that we can measure that using PET scan. So sorry for the slide in French. So you can use it to use PET scan to look at the difference between the different states, and also related to this perception of stimuli. So if you look at the response, for example, in the primary auditory cortex, some sound. So you don't have here a difference between people in wakefulness and people in anesthesia. So you've got the same peak. But if you go in the next area, so the association area in the auditory pathway, then you've got a clear difference between people awake and anesthetized. So you've got a difference in the propagation of the signal in the network for this kind of data. And actually, this kind of method has been extended to monitor the state in terms of conscious state of the patient. So you can apply TMS, for example, so transmagnetic stimulation to the brain, to some areas, and see how this stimulation propagates in the network. So here you've got different areas. And you can measure the complexity of the signal. So you can see here that for people in the vegetative state, so deep coma, they have hardly any response, just locally, and not in the rest of the network. And if you look a bit later, there's partial recovery, and then you start to have more response, but they are still quite similar across responses. And then later, with better recovery, then you start to have more heterogeneity and a broader network that is activated there. So this is what I mean by complexity. Here is how diverse this response propagates in the network. And that's a hallmark of the conscious state of the subject. So the data that we had there were data from coma patients. So when people arrive in the service in Liège, so they usually had accident or can be stroke or anything. And then they're in the so-called unresponsive state, so deep coma, and they don't really interact, they can't respond. They can't respond to stimuli by the staff. And then after a few days, a few weeks, they can recover and have minimal consciousness, which means that they can have some limited but still interaction with the staff. And so we had also a cohort of healthy control for comparing with them. And so when we applied effective connectivity to these three groups, so we look at the differences between AWS and healthy control and MCS to healthy control. And to our surprise, what we found is a mixture of increases of effective connectivity and decreases here for this patient, and here only decreases. So only significant interaction are shown here, significant differences. But here it's quite counterintuitive because you expect the minimal consciousness to have very low level of activity. So why would you have some increase of interactions there? So to understand that, we had to not analyze this result like interaction by interaction, but collectively. Okay? And so what do we mean by collectively is when you look at an example network, like here, you can have a change, increase or decrease. So here I'm just showing an increase, for example, of interaction. But you can have some changes at the node level. Okay, so here, all this connection would increase, meaning that this node here would be a broadcasting hub. So it's going to send more information to the network or more activity at least. If in this case, so this node is, this hub is going to be listening to the network. And so that's some changes related to one node. And on the other end, you can have changes at the network level. So here you've got these two interactions that increase, and that's going to make a favored pathway for the propagation of activity in the network. And if you look at this, you've got a loop, and that's expected to further reinforce a pathway that determines a community where you've got stronger activity, right? So when you've got increase and decrease in our network, we want to go and analyze to try to discriminate this different situation. Because of course, if you've got something like this with increase of interactions and weights, we expect to have potentially a sustained activity on the long term. So is it what's happening with the UWS patients? And so to do that, we use the same network that we fitted to the resting state fMRI of these patients. And we're going to use in silico perturbation, as said. So it's just stimulating the model and look at the propagation of the activity in the network. Okay. So we're going to use a network that's been trained on ongoing activity that perform the stimulation in it. And so what we can do, so that's some framework that we developed, especially with Gorka Zamora Lopez. So that started in Barcelona. And so the idea was to transpose network theory to dynamical systems. So if you look at here, you've got a representation of the effect of stimulation over time. So early time after the stimulation, you just have, so if you stimulate here, you will have just the node 2 that will be that will have an increase. So because you've got a direct connection between 1 and 2. If you stimulate 2, it will be 3. If you stimulate 3, that will be 4, and so on, because you've got a loop. But if you look at the propagation of activity a bit later, then you've got the network effect starting to come, and in the end, you've got a very homogeneous response. So here for each node, or globally, if you stimulate all the nodes together. And so with this, you can quantify the network response either by stimulating one node. So if you look at these matrices this way, or how one node is listening to the network if you look at it horizontally. And so in the end, if you make summations of these matrices, you can have the receiving and broadcasting level of each node over time due to response. Okay. So why this is nice, because it's directly related to some measures in network theory. So with this, you can look at different nodes and see whether some nodes there are more listeners or more broadcasters here. And you can compare that to, so you can define hubs in a dynamic fashion and compare that to hubs that you can define using structural connectivity or functional connectivity. And what we see is that, in a sense, these dynamic hubs, they are not necessarily the same as the SC and the FC. So it's complementary information, so to say. So we use that for our 3 groups here, healthy control, UWS, and MCS. So looking at these matrices that you saw before. So here, that would be stimulating one node, here another node. And that's the result that we see for our 3 groups. So you can see here that you've got, for UWS, along this diagonal, you've got stronger values. So that's for the very early stage. But when you look at the late stage compared to the healthy control here, so then you've got a decrease of response. So if you sum all the elements of the matrix, you've got this curve here in red for the UWS to be compared with the healthy control in black. And so you see that you've got a strong overshoot in the first stage, but more importantly, you've got a decrease on the long term. And this long-term response is responsible for quantifying the integration in the network. So how this perturbation, they propagate in the network. So we find that this UWS, they've got a lack of long-term integration that is partially recovered when you look at the MCS in blue. And so the increase that we saw in the UWS, they're actually only responsible for this very early overshoot that kind of correspond of an over-segregation. So you've got maybe compensatory effects that have disconnect that comes after the well, because of the non-responsive state that cause region to kind of cluster, but this region by clustering they can only talk to themselves and they are disconnected from the rest of the network. So that's why you got this overshoot here. And the last bit that we analyzed there was to look at the changes in region that were receiver or broadcaster. And we look at differences between UWS and healthy control. And so that it was posterior and thalamo-frontotemporal circuit that were mostly affected in broadcasting and receiving for the UWS. So that's one way also to give potential therapeutic targets for the clinicians to understand when which region would lack some dynamic properties and some region that could be stimulated to favor or to help the patient to recover from UWS to MC. Okay. So that was it for this coma patients. And another study that is more recent focused on a different conscious condition, which is sleep. Here we've got data from Taliazuki that recorded in fMRI subject, healthy subjects this time, and from wakefulness to sleep stages. And so here we've got the different sleep stages from light sleep and 1 to deep sleep and 3. And so when we fit our model to the data, we can look at the difference between the effective connectivity across the different stages, and we see some changes, but not so strong. We look at, so here you've got the connectivity, here you've got the input variance. So globally, if you look at these parameters, average over the whole network, you don't see much of a difference, but when you try to use them to decode the stages, you can see that connectivity and the input covariance, they are in fact quite informative. So here is the decoding accuracy compared to chance level in gray. Okay. So the question here was how to analyze this data. So if the change across the subjects, the stages, sorry, is not in global properties of these matrices, like what one could expect is, okay, for awake, you've got very strong, much stronger connectivity. For deep sleep, that would be quite low. But that's not really what we see. So to analyze that, we went for another framework, which is entropy production. And so that's a nice framework coming from physics. And actually, for this simple network of the Stein-Beck, there is an analytical manner to calculate that. And so when we look at the entropy production this time across the stages, we saw a kind of gradient between them. So it's not strikingly changing, but we see that we've got more entropy production for the wake state compared to the deep sleep state. And we can not only look at that entropy production at the level of the whole network, like here, but look at all different regions here, and we see that in terms of entropy production, most of the regions here, they lowered their entropy production, and only a core of a few regions, they remain active, like this one and this one. Among them, precuneus, visual system, and so on. So there's just a few core regions that remained in the high state of entropy production. So when you look at now the data, the parameter that we estimated from the network, we saw that for changing the contribution of the connectivity, the time constant of the system, and the input covariance were contributing to this entropy production, but the relationships were not so clear. Like here, it's not the global level of input covariance that matters, but that's more the detail of its structure. And so to explain that, because that's quite not obvious there, so let's go back to a bit of mathematics. So I just recall our dynamic system here. It's Jacobian, meaning the connectivity and the time constant. Our inputs that are beta here. And so to understand whether you've got entropy production, it's like considering whether the time courses generated by this dynamic system, they can be time reversible. So that's some properties in physics that are well known. And so for this, Einstein and Beck, this corresponds to a commutativity condition on the model parameters. So if you've got the matrix J, so corresponding to the connectivity, and the input covariance that commute in this way, so with the transpose, then you've got time reversibility. If not, you don't have that. So what does that mean? That means first that it's not a global property of just if I scale the two matrices, that won't change this commutativity condition. So it's really the detail of the structure of how regions are connected and how much noise they receive that matters here for this entropy production. And so to give you a bit of an intuition, so if I've got a network like here with two nodes and balance interactions, they both have the same level of input variance. And so if I look at my system, so the commutativity condition that I showed before relates to this Onsager matrix that's defined here, L. And so I can analyze it with its symmetric part and this anti-symmetric part here. So what you have is represented in this graph, so it's a bit abstract, but basically you've got the two activities, x0 and x1 here. So this ball, the blob here in black, correspond to the shape of the variance, the input variance. So here the two nodes, x0 and x1, they receive the same variance. So you've got a spherical blob. So it's a Gaussian here. And so these arrows, they correspond to the effect of the interaction. So basically what you have is, if you think of the activity in this space, it's going to diffuse around the distribution. So it would be a random walk in the distribution determined by the blob here. And the arrows, so the interaction, they are pushing this, the activity back to the center. So that's why you've got a stable system. And in a sense, if you look at, if you simulate over time, you will see a kind of random walk that being always pushed to this center. And so this decomposition in symmetric and anti-symmetric parts tell you about the time reversibility. So if you've got some arrows here, so this is now the combined effect of this diffusion due to the input and the drift due to the interactions. And so that is shaping, because you've got excitatory rates here, you've got correlation between the two variables. So the distribution of the activity of the two variables is being shaped here. So it's elongated. You can see that you've got positive correlation between the two variables, as expected. And so the symmetric part here is pushing the activity back to this stationary distribution. And you don't have anything for the anti-symmetric part. So what's happening when we do a stronger connection from one region to the other? So that's not going to change the stationary distribution, as you see here. But what we will have, in addition to this phenomenon here, which is time reversible, is a time non-reversible rotational movement. So it's somehow the interaction between the drift here and the shape of this field that causes this rotation around or in the stationary distribution. Okay. So the intuition is that because the direction of this diffusion is not aligned with this, the effect of the interaction, you end up having some kind of rotation, and rotation in this network means the preferred path for propagation. Okay. So that's what happens when you've got stronger effective connectivity weight from one region to the other. If you have positive and negative, some inhibition, that's even stronger. Okay. But what's not so trivial is that when you have stronger variance for one input compared to the other, even if you have the same interaction between them, you will get this rotational invariance. So somehow what this describes is simply that, which is maybe a bit trivial, but here the cool thing is that it's quantifiable, that when this node receives stronger input, it's going to drive the other one, and you're going to have this rotation in the network. So that was maybe some long mathematics to explain you something that is kind of trivial for to know, but it's something that really scales up at the whole network. And with this, you can quantify pathways for favored propagation in our complex network. And the complexity is really coming from the data, from the fMRI propagation dynamics. So I think that's a nice thing to describe from resting state activity. You've got some waves that go in the direction of, in some specific direction in the network. Okay, so that was the last part of the mathematics. I hope it was okay. So maybe I just have 2 minutes to finish. 3, 2. So I just wanted to talk about functional modeling. So we talked about connection-based model yesterday. Here I was focusing on effective connectivity. So effective connectivity, and we also saw some things about simplices and analysis and directed network a bit. So somehow this effective connectivity is going in that direction, not with binary networks, but with some networks that are directed and weighted. And so this is our key signatures for the brain dynamics. And here, so the way we use this anatomical information, so this connectome, in the network is just to constrain the topology of our network. In contrast, you've got some other models, like usual connectome-based model, there's a virtual brain, there's a dynamic mean field, or the Hopf model from Gustavo Deco, for example. And here, so they use this connectome, so the DTI data, to determine the strength of this activity in the network. And so instead of changing the weight here, so they are fixed weight, but they tune the regional dynamics. Okay, so as I said, virtual brain and dynamic mean field about that. And the key is how you make a biophysical model of your dynamics and how this affects the activity pattern that you can quantify this functional connectivity. And of course, the point is to feed that to empirical data. So I just wanted to argue that so this line of modeling started with studying the link between structure and function, so how regional activity, biophysical properties of the model, they can impact the activity at the global level. But this has led to a bias, in my opinion, on the focus on goodness of it. So the point for a long time has been how good my model can reproduce the data and how complex my biophysical model can be or simple can be to explain different aspects of this network pattern. But with the effective connectivity model, what you are doing is really using this estimated effective connectivity as a representation of the spatio-temporal structure of the signal. And so we can use that for biomarkers, and I think that's a powerful use that is not so common here. So here you could also have biophysical model, estimate some parameters, so let's say one parameter per region. And so in the end, these parameters here and these parameters here, for the same data, they could be different representations of your spatio-temporal signals. And so the key difference here is that here, if you got one parameter per region, you've got, let's say, n region, n parameters, but here you would have at the order of n squared. And so you've got richer biomarkers there, or richer signatures, higher dimensional. So more difficult to estimate maybe, but potentially more powerful to explain the network pattern. And the last bit that, if you're interested, I can comment, but in terms of effective connectivity, there's not just the version that we had, but the most famous version is DCM. And in fact, we showed that for resting state DCM, we've got an alignment between our method and their method. So if you look at, let's say, 50 subjects, you've got an alignment, Pearson correlation between each individual, between the group average that starts to be 0.5 or above. So you really have aligned measure from our version of effective connectivity and the regression DCM. And you can even make transfer learning, so train some decoder on one version of EC and decode for the other version. And so that's just to, for those who are interested in effective connectivity. And I guess I'll skip that, just mentioning that we also, so we also working on some acquisition of clinical data now in Marseille. So for example, with amyotrophic lateral sclerosis. And so there we are trying to combine different modalities of fMRI. So T1, so quantitative T1, that's my usual. From diffusion MRI, you can get the fractional anisotropy here, but you also have sodium MRI that correspond to this map, so they're a bit blurry, but they capture, in a sense, the metabolism, so the imbalance between sodium and potassium in the neuronal tissue. And so you've got this that can capture potentially alteration in the metabolism that come before the degradation of the neuronal tissue that can be reflected in the fractional anisotropy or the quantitative T1. So we try to use that to predict the evolution of ALS patients, which is quite devastating disease. And so what we found is that sodium and fractional anisotropy here are the informative features. So the point later is to try to design some models, so it could be some connectome-based model, where the biophysical model would incorporate the microstructural alteration quantified by FA or the metabolic effect with the total sodium concentration. So the point is to find a mechanism to describe it mathematically to be able to combine these different things in a meaningful way. And another thing that we are doing, for which we need a next generation of an functional model, is psychiatric disorder. So we are working so far mostly on bipolar disorder in Marseille. We've got a neurodevelopmental hypothesis. And we've got structural, functional MRI, also have transcriptomics about patient and so on. So the idea is to try to make models and to see whether we can combine this effective connectivity with this biophysical modeling in a single model. And with this, just recapping a bit what I said about this anatomo-functional model, trying to make a balance between predictability and interpretability. And so, yeah, the way I see it is that so far we've got very blurry map because we don't have my tools. We've got limited prediction and things like that. But let's go on and keep sailing because the world is vast, and the more we discover, the more we are going to be able to make better prediction, increase your tools. So the travel, the journey is maybe more fun than the goal to reach. And I thank you and thanks all the collaborators, because this was, of course, collective work with many people that contributed to this work. Thank you.